Hey, good morning. I hope all of you enjoyed your long weekend. So let's continue talking about cellular respiration. That is how cells utilize the energy that's stored in glucose and converting that glucose into ATP, right, which is the chemical form of energy that all of the different biological pathways in the cell, in particular, all the enzymes that run these different pathways in the cell, are expecting as the form of energy so that they can actually perform their functions. So far, we've been talking about the process of glycolysis. Today, we will continue on to understand how, as life evolved and as the atmosphere of the Earth changed billions of years ago, there were additional options made available to get more energy out of one molecule of glucose. And so that will lead us into our discussion of aerobic respiration and taking a look at the citric acid cycle. Okay, so you'll recall that when life first evolved on Earth, so Earth is about 4.6 billion years old, right? In the first billion years of the Earth, as it's cooling down, there was no life, right? So life started about 3.5 billion years ago. And as we've been talking about, at that time, right, there was very little atmospheric oxygen. Absence of O2 in the atmosphere meant that primitive forms of life, prokaryotic cells, single-celled organisms, had to make energy or make glucose through photosynthesis because there was plenty of sun shining down on the Earth, and that represents energy. And there were plenty of compounds like CO2 and water available in the primitive Earth's environment, right? And so photosynthesis, as we will talk about more in depth later, involves those early organisms harnessing the sun's energy and putting together carbon dioxides and waters into glucose. Right? And then that glucose with its 6 carbons represents a lot of potential energy. Each one of those carbon-carbon bonds, right, represents electrons that are sort of stably in these orbits, and those electrons would happily, if given the chance, right, break from those bonds and reform other bonds with other atoms at lower energy, right? And so therefore, glucose was a good raw fuel source molecule. Right, the sun's energy put into the bonds of making the glucose, and then those individual bonds of the glucose could then be broken when needed to get some energy out, and that energy could then be converted into ATP so that the ATP could then be distributed throughout the cell, and the ATP would then be bound up by different enzymes that would then break the ATP, right, to get the energy out of it, right, in order to do their enzymatic functions. So we said that because there was no atmospheric O2, the process of breaking down glucose to get ATP from it had to happen anaerobically, right? Anaerobic means the absence of O2 being involved. And as we studied the last couple lectures, that goes through a process that we call glycolysis. So in the cytoplasm of the early cells, and importantly, in our cells as well today, right, still start off by breaking glucose down through glycolysis. And through that process, you end up with getting a couple of ATPs out of a glucose molecule, right? And you end up with these 2, 3 carbon molecules left over. These are the pyruvates, right? So remember, pyruvate is the end result of glycolysis, right? And early life didn't really have any real way to do anything more with pyruvate in terms of breaking bonds to get more energy out of it, because that's just how it evolved. You ended up being stuck with pyruvate, and that was it. Right? But as we learned, primitive bacteria and other microbes learned, well, at least you can use the pyruvate to empty out some of those electron-carrying coenzymes, like the NADHs, that ended up getting electrons put into them, right, during glycolysis. So remember, during glycolysis, during the process of making a couple of ATPs from a glucose molecule, you have to transfer some electrons out, right? And those electrons are transferred by the enzymes that are working on the glucose, and they transfer the enzymes into coenzymes like NAD+, which serve as convenient little baskets, if you want to think about it that way, to carry those electrons away, so that the enzymes themselves aren't stuck having to like hang on to these electrons. Right? So fermentation, as we learned, is the process of taking the pyruvate and doing something a little bit more with it, not to get more ATPs out of it, because you cannot. Without more available oxygen available to help take electrons, you're kind of stuck, right? But at least what you can do is you can use the pyruvate as a way of sort of dumping those electron-carrying baskets and freeing them out. So the NADH becomes NAD+, so it can go back and help out with glycolysis, right? And different enzymes like lactate dehydrogenase do that to the pyruvate, right? They transfer the electrons onto the pyruvate, reducing it into lactate. Right? Or certain microbes can take the pyruvate and through a series of a couple of different enzymes, convert the pyruvate to acetaldehyde and then to ethanol. Again, as a means of simply emptying those NADH electron-carrying baskets so that the empty basket, the NAD+, can go back and help keep the glycolysis going. Right? So the fact that you only get 2 molecules of ATP from a starting molecule of glucose through glycolysis, that's pretty sad, right? I mean, that represents only about maybe like 3% of the available energy in those carbon-carbon bonds in the starting glucose that you're actually successfully converting into a couple molecules of ATP that can then float off in the cell, right? But you're stuck with that, right? But right, you're stuck with this because, again, right, in the absence of available O2, atmospheric oxygen, you really can't do anything more than this, right? But what was happening? That first billion years or so of life evolving, right? So now from maybe 3.5 billion years ago forward, right, as photosynthetic primitive organisms are putting together carbon dioxides and water to make their C6H12O6, their glucoses, right? They're left over with a molecule of O2. Right? And that gets lost into the atmosphere. Remember that nice painting I showed you of a primitive ocean, right? Where you have these photosynthetic like bacteria sort of growing on the bottom of the ocean, and there are these bubbles of oxygen coming out of them, right? As they're busy putting together glucose. Right? So slowly but surely, the atmosphere of the Earth began to fill with this oxygen that really is the waste product of those microbes making their sugars, right? And as suddenly the levels of oxygen began to build up, right, now you have O2 to kind of play with, right? And as we said, O2 is really important because oxygen is a really good electron acceptor, and suddenly that means you have additional options, right, for getting more mileage out of your glucose than just being stuck with turning it into pyruvate. And getting a couple of crummy ATPs out of it, right? With the availability of oxygen, you can now take this pyruvate and really work on it some more to get the electrons off and end up getting way more ATPs out of it, right? So that's why the development of aerobic respiration, importantly, only happened after enough oxygen was building up in the atmosphere, right, to be able to motivate or encourage the evolution of new pathways to be able to use that oxygen to get more energy from the glucose. Right? And the other important thing I want you to think about, right, is these options of using oxygen, right, are added on after the process of glycolysis happens. So evolution, oftentimes, when evolution works, it develops some complicated mechanism for accomplishing something like glycolysis. Even though things change and you might have way better options, compared to just being stuck with glycolysis, right? Evolution cannot just simply give up glycolysis altogether and adopt something new, right? Because evolution is a gradual process, right, of adding things on or things changing. I mean, think about it. You would never imagine that at some point in early evolutionary history, there's a lot of oxygen in the atmosphere now, and suddenly these anaerobic bacteria, right, that only know how to do glycolysis are suddenly going to be like, all right, forget about the glycolysis, let's do something with the oxygen, right? Because you cannot, right? So just like, how evolution of certain things that we use in our daily lives, we're sort of stuck with them as sort of baggage. Right? A good example is, for example, when you think about automobiles, right? Hey, we all are familiar with driving cars, and we're used to a steering wheel that just kind of turns right and left, and then we have to worry about with our feet, gas pedal and brake pedal, right? Because from the earliest days of having cars, like Henry Ford and his Model T car back in like the late 1800s or whatever, right? That was the design, right? You had to have some way of steering, and you have to have some way of doing acceleration and brakes. Right? So these days, I think there are many modern experimental car designs try to have things like the steering wheel be also where you control things, acceleration and brakes, because they've done lots of studies to find that if you just do everything with your hands and not have to coordinate it with your feet, right? Like you can push forward to accelerate, pull back to brake, sort of like airplanes, right? Airplanes have sort of a mechanism that's all in the steering wheel, right? So that's actually a lot more efficient. But the problem is, because over the last 100 years, all automobile manufacturers, right, design cars around this idea of a steering wheel is just for steering and brake and accelerating pedals are worked by your feet. It's very hard to just scrap all of that, right? And imagine, oh, next year's models of cars all have everything done by the steering wheel, right? Because it would just be too much of a change, right? Things have to happen incrementally. Right? And that means for some time, we're going to be stuck with cars being the way they are. So I use that example because that's what we're talking about here, right? If life evolved on Earth, depending upon glycolysis first, right, then even though later on you have the ability to do aerobic respiration, right, life on Earth could not just simply scrap the glycolysis part. What it could do is add onto it. So to this day, the cells in your body, right, and the cells in all living things that perform aerobic respiration, right, we perform aerobic respiration, do it as an add-on to first doing glycolysis. Okay, so that's very important to remember. Your cells first do glycolysis and then they do aerobic respiration. So what does that mean? That means from pyruvate that's generated in your cells, right, you don't really worry about going down the pathway of fermentation in order to keep, glycolysis going. What you want to do is you want to feed that pyruvate from glycolysis into an aerobic respiration pathway. And that first has to do with the conversion of pyruvate into this molecule, acetyl-CoA. Right? So glycolysis ends with pyruvate, right? But now, because your cells have a lot of oxygen available, right, now we're going to talk about what happens next in your cells. Right? Because by utilizing oxygen, you can get a lot more ATP out of the glucose this way. So the first step is converting the pyruvate. So remember, pyruvate is 3 carbons because it represents half of the starting glucose molecule, and each half has been worked on now through glycolysis, right? And so you end up with this pyruvate molecule. Pyruvate is converted into acetyl-CoA. So you can see that one of the carbons is released from it. If you look at it here, you can see the pyruvate has 3 carbons. This carbon is actually a carbon bound to 2 oxygens, right? So if that were to break off, that's CO2, that's carbon dioxide, right? And you're left with the remaining 2 carbons here. So that's what you have here, the remaining 2 carbons with the oxygen double bonded to it, right, over here. And you add on this coenzyme A, right? And this molecule, this acetyl-CoA, is what feeds into what we call the citric acid cycle, right? Because this acetyl-CoA will form the basis of generating a lot more electrons that will end up powering ATP synthesis. All right. So let's just kind of summarize what we are talking about here. We said that all living cells, right, all living things on planet Earth perform glycolysis, right? Whether that's all they do, like anaerobic bacteria, right, and they just stop there and they make lactate and ethanol, for example, as their waste products of glycolysis, or, for example, our cells that still start off with glycolysis, but now we feed the product of glycolysis into an aerobic respiration pathway that's tagged onto it, right? So in the cytoplasm, this is where glycolysis happens, and you get very modest ATP yield, right? You only get 2 crummy ATPs made from each glucose, and you end up filling some of these NAD plus baskets, right? These coenzymes that carry the electrons away so that the enzymes that are actually working on the glucose aren't stuck with the electrons themselves, right? Because remember, if they were stuck with the electrons themselves, right, then you'd have to have another enzyme that comes and takes the electrons off of that first enzyme so that the first enzyme can keep working. And then what are you going to do with the second enzyme? You need another one to free that one up, right? So it's a lot more efficient if you just have things like NAD+, these little baskets that are bound to the enzyme at the same time that the enzyme is working on its main substrate, like the glucose intermediate molecule. And when it transfers electrons off of that, it just puts them in these coenzyme carriers, and the NAD+ becomes NADH, because remember, not only electrons come over, but the electrons have protons attached to them, right? And then these baskets can then float off. Right? So as atmospheric oxygen began to build up on planet Earth, life evolved to make use of that. So the conversion of pyruvate to acetyl-CoA would be the important second step. And that acetyl-CoA then feeds into the third step, which is the citric acid cycle. So what's the purpose of the citric acid cycle? You know, you might say like, yeah, I've learned that before. It's to make lots of ATP, right? Yeah, but that's like the super simplified version of it, right? Yes, the citric acid cycle's function is to eventually support the production of lots of ATPs being made, or lots more ATPs being made from the starting glucose. But the citric acid cycle itself is not about making ATPs. The citric acid cycle itself is about generating lots of free electrons that are coming off or being taken, right, as the intermediate molecules of the pyruvate to the acetyl-CoA and the acetyl-CoA being processed, right? Those bonds are being broken, right, and electrons are being taken off. Those electrons are put into more of these electron-carrying baskets. So you have lots of free electrons that are being generated that can now be kind of shipped off somewhere in these coenzyme baskets. And those electrons, right, in step 4, as we will learn not today, but in the subsequent lecture, those electrons power a series of membrane pumps, right, that pump protons across the membrane to build up a proton gradient. Right? So this is exactly what we've learned before in terms of active transport, building up big gradients of things like protons from one side of a membrane to the next, so that you can allow the protons to flow back down the gradient and power some other types of membrane proteins, right? So it's those electrons that are generated by the citric acid cycle that will power what we call this electron transport chain of proteins. And that will then create a proton gradient that ultimately will be used to create many ATP molecules in sort of a mass-produced way. Right? Where does the O2 fit into this, right? If I said that from steps 2, 3, 4, and even though I don't show step 5 here, the actual manufacturing of the ATP, we will get to that. You know, I think you're thinking like, yeah, so the whole theme is these subsequent steps could only happen because of the O2 that's available now, right? But where does that fit in here? It fits in because the O2 is such a good electron acceptor, it's greedy for electrons, that the O2, the presence of O2 is what's going to drive the direction of these electrons flowing, right? O2 is available to receive these electrons, doing things like encouraging the emptying of these baskets, right? So we'll get into that in time, right? But let's go ahead and take a closer look at these steps 2 and 3, acetyl-CoA and the citric acid cycle. First off, right, I've been emphasizing this idea that in order to get a lot of ATPs produced from the glucose, you have to think of it almost like mass production, right? Like a factory that's creating lots of ATPs in a mass-produced way. This is compared to the way that you can think about the ATPs are being put together during the glycolysis, right? Glycolysis, as we've been learning, right, is this multi-step process through 3 main sort of phases of glycolysis, where the glucose is essentially rearranged, it's broken into 2, and then each half is then worked on, right, by a series of enzymes. And at a couple of steps along the way, certain enzymes will take an ADP, right, adenosine diphosphate, and actually hold on to it and transfer a phosphate group from the intermediate glucose, the molecule, onto the ADP to make ATP, right? So that's a process that we call substrate level phosphorylation. In other words, imagine this purple thing here is one of those enzymes in the glycolysis pathway, right? And at this particular step of glycolysis, this enzyme, remember, it's a protein, so it's the right shape to bind a couple of substrates at the same time. In this case, one of the substrates is ADP with its 2 phosphate groups attached to it, right? The other substrate is this 1,3-bisphosphoglycerate. Right, so this is one of the steps in glycolysis where you have a 3-carbon molecule with 2 phosphate groups attached to it. And the enzyme brings these 2 together, right, in such a way that it rapidly accelerates the chemical reaction where the phosphate group comes off of the 1,3-bisphosphoglycerate and gets added on to the ADP, therefore making ATP, right? So the enzyme transfers the phosphate from a donor molecule to ADP. That's why it functions as a catalyst, right? In other words, a molecule of ADP floating around in the cell and a molecule of 1,3-bisphosphoglycerate floating on the cell, could they by chance crash together in just the right shape so that this reaction happens on its own? Yeah, right? But hey, what are the chances of that happening? How long are you going to wait for that to happen, right? That's why if you have an enzyme that binds both and is the right shape to shove them together, you accelerate this, you catalyze this reaction potentially hundreds and thousands of times faster than if these 2 molecules were left to kind of float around uncatalyzed, right? Yeah, that's what enzymes do. That's why enzymes are so important. But consider this. We call this substrate level phosphorylation because it's like hand making each ATP, right? You can sort of see that, right? It's almost like you imagine if you were to actually make something with your hands, right? Yeah, you can do it, but it's not very efficient, right? So the pros are it works, but the cons are, hey, it's really slow, right? And that's part of why glycolysis alone only ends up generating 2 ATPs. So if you wanted to, take this thing that you're making, right? Let's say you're a little home entrepreneur and you make something with your hands, it's your little prototype, whatever. And now you want to sell it, you want to go big time. You want to find maybe a factory that can make this thing in a much more mass-produced, high throughput kind of way, right? That's where all of these electrons come in. I was saying how the citric acid cycle that we'll learn about has to do with generating lots of electrons, right? The electrons can be used to kind of power, for example, setting up a proton gradient, right? And then allowing the protons to flow back down through the membrane, from high to low after they've been set up by some sort of set of pumps or whatever. That can be your power, right, to mass produce things. So that's what we're discussing here. So the electrons, as we will learn, right, are used to sort of power or encourage a series of different proton pumps in the membrane, right, to pump protons from a low concentration to a high concentration, right, against their natural flow. Once you've built up a huge gradient from one side to the next, you can then tap into that by allowing the protons to flow back downhill through these other proteins, right? And if these other proteins are designed the right way, right, the movement of the protons through them will make the proteins move, right? And the movement of these proteins can then do sort of like a mass-produced form of adding phosphates to ADPs to make ATPs. Right? So it's kind of like in this photo here. If you have a big crowd of people, you know, on one side of this set of turnstiles, right, and then you have them all flowing through the turnstiles. Each one of them is kind of pushing the turnstiles. If you could harness that, you could actually generate, right, some energy to do some work. So if these people are protons flowing through here, right, through these, the lighter or this colored purple pump here, you can imagine, right, that as they are making the protein move, if the protein is designed the right way, you can make a whole bunch of ATPs, right, much more rapidly high throughput compared to your sort of sad hand making the ATPs during glycolysis. So the pros of this are that you have very efficient production of ATP, but the cons are what? Well, you have to set up this proton gradient in order to let the protons flow through to power these ATP synthase proteins, right? And so that requires a steady source of electrons going through these proteins over here in order to keep this proton gradient built up. Right. Okay, so where does this happen? This all happens in the mitochondria. Right? And it involves several examples of different types of ATPases or ATPase pumps. We've learned about previously, so this is an old table from chapter 8. We learned about, oh, there are ATPases that are P-type ATPases. They work by breaking ATP, getting a phosphate added to them, and that changes the shape of the membrane protein so that it can do work, right, push things against a gradient. Some of these are V-type ATPases. That was the example of like the gumball machine that I showed you previously. There are other types of ATPases that move different types of toxins out of the cell, right, to detoxify the cell. The one that we're going to talk about now are these F-type ATPases. Right? So these F-type ATPases all involve the movement of protons. Okay? In eukaryotic cells like our cells, this takes place in the mitochondria. So I don't know how many of you are familiar with this sort of theory that eukaryotic cells evolved from prokaryotic cells. Well, I think you know that, right? Because all life on Earth began as prokaryotic cells. But where did eukaryotic cells came from? You know, it's believed that after the first, you know, billion years of life just being prokaryotic cells, like this is like 3.5 to maybe 2.7 billion years ago, you began to have some larger prokaryotic cells actually taking up smaller prokaryotic organisms, like primitive bacteria, things like that, into them. And not to destroy or digest them, right? But just, you know, by mistake, you know, the larger prokaryotic organisms might take in smaller ones, and they might actually coexist very happily that way in this kind of endosymbiotic relationship. Right, where the small little prokaryotic cells that got taken in, hey, this is a nice home. They're protected, right? They have access to nutrients. And in exchange, the larger prokaryotic cell that took the smaller ones in benefit from stuff that the smaller ones are producing inside of it, right? And so it's believed that the formation of all of the organelles that we learn about now in our eukaryotic cells, right, is a function of this happening very early in single cell evolution. So the mitochondria itself is believed to be, you know, what's left over from early prokaryotic organisms that were taken in. And in this case, the early small little prokaryote, right, its ability to generate ATP, right, is then used by the larger cell that that had it. And so over eons of evolution, of course, you stop having this concept of, oh, it's a large cell organism with smaller cell organisms in it, right? Over eons of evolution, billions of years since then, right? The mitochondria have ceased to exist as a separate organism. They are now part of our eukaryotic cells, right? Okay, so that's why in eukaryotic cells, like our cells, right, this type of ATP synthase is happening in the mitochondria. In prokaryotes like bacteria, yeah, I mean, they are themselves, right, a prokaryote. So this is just happening on their plasma membrane. And in plants, these happen on the thylakoid membranes of chloroplasts, right, that are inside plants. We'll study that when we study photosynthesis. In any of these situations, what we are talking about is using a proton gradient. So let's zoom in here. So here's a mitochondria. You can see here the mitochondria has an inner membrane and an outer membrane that creates basically two different spaces inside of it, right? This inner space, and then between the inner space and the outer wall of the mitochondria, you have this kind of outer space, right? And so you can see here, this inner membrane is studded with these different types of proton pumps. So let's look on the left. Let's zoom in on this little cross section. So in this case, the orangey regions over here and here would be the inner mitochondrial space. The more pinkish, kind of darker space over here would be the outer mitochondrial space. You will have certain proteins like this purple blob here that are pumping protons from the inside to the outer space, right, building up that huge proton gradient, right? So you'd have maybe 100,000 times stronger proton concentration in the sort of darker pink space over here compared to the inside. And then you would have other proton pumps, right? Like this purple one here, that will allow the protons to flow back downhill, right? Because they want to so badly, right? Go from high to low back into this inner membrane. And you can couple that then to the movement of these purple proteins to make ATPs, just like the turnstiles of a huge crowd of people entering something like a stadium for a sporting event. Okay, importantly, these are still called proton pumps, even though they're allowing the protons to flow downhill in this direction. Why? Right? Because they're going in this direction simply based upon the gradient of protons. Right? These pumps, just like many other examples of enzymes that we've studied, can go in two directions, depending upon how they're being motivated or pushed. Right? In other words, if you had way, way, way too much ATP in here, right, and very little protons out here, this pump would work the opposite direction. It would actually break ATP to ADP as part of pumping protons in the opposite direction. Okay, but because it's set up so that it's oriented this way, and you have these other proteins creating a strong proton gradient in this direction, right? This then favors the protons flowing back down through these ATP synthases in this direction of the arrow. And so it favors then the direction of this reaction happening, ADP to ATP. So this cartoon here, I love this cartoon. You know, there's not many cartoons that I really love in our textbook, but they did a really good job with this one. I know you're looking at this going like, oh my God, what is this? Do I have to memorize this, right? No, you don't have to memorize it, but I want you to kind of use this as the basis of you piecing together these different things that we're going to learn about from last week to this week to next week, okay? Because this cartoon does a really good job of it. All right, so what we're looking at here is we're zoomed in on one section of a mitochondria, right? So that's why here, this big thing, this is the wall, outer wall of the mitochondria. This tan kind of part is the cytoplasm of your cell. Right? And then, of course, this pinkish part here is inside the inner membrane of the mitochondria. So remember, right, you start off with a molecule of glucose that's been taken into your cell by something like the GLUT1 transporter. And so the glucose has been delivered. It's like the coal being delivered to the factory, right? It's your raw fuel. So it's got to be converted into ATPs so that the proteins in your cell can use it. Right? So remember, the first step is glycolysis, right? Even though we're so much more advanced than primitive bacteria 3.5 billion years ago, hey, we're saddled with this baggage of that's how life evolved, right? So that's how the cells in our body to this day still start off using glucose. I think that's kind of neat, to be honest, right? When you think about it that way. But anyhow, so the glucose right, is broken down through glycolysis. You get a couple of ATPs out of it. They don't bother labeling how many ATPs in this cartoon, because that's not what this cartoon's about. It's just a reminder, you get some ATP just from glycolysis, right? And you end up making pyruvate from it. So our cells, if there's plenty of oxygen around, right, we won't favor the conversion of pyruvate through fermentation to lactate, right? That's what happens when your muscles don't get enough oxygen and they don't have a choice, right? They end up sort of favoring the production of lactate in order to free up more electron carrying coenzymes to keep the glycolysis going, right? So you end up with pyruvate, and the pyruvate is then transported into the mitochondria. Step 2 is the pyruvate is oxidized to be made into acetyl-CoA, right? And then the acetyl-CoA is going to be the starting point of the citric acid cycle. Right? So we will look at this in more depth in a couple of slides, but for now you see the citric acid cycle. It does produce some ATP, but again, it's pretty wimpy, the production of ATP, because that's not really what the citric acid cycle is about. What is the citric acid cycle about? It's about taking apart the acetyl-CoA as part of other intermediate molecules and being able to get electrons out of these molecules to fill up more of these electron carrying baskets, right? The citric acid cycle is all about generating free electrons that can then be shipped off somewhere else, right? So you see NADHs. So even through glycolysis, you make a couple of NADH baskets that are carrying electrons. Right? The oxidation of pyruvate, you fill up a basket of NAD to make it NADH. Citric acid cycle generates more of these filled baskets, not only the NADH, which is the nicotinamide adenine dinucleotide, but also we'll look at, there's another one, this flavonoid electron carrier here, the FADH, that also carries electrons. And look at where all of these baskets, these filled baskets of electrons go. They go to power this series of brown proteins here, right, that are set up on the inner to outer membrane, right? This orangey membrane divides the inner mitochondrial space to the outer mitochondrial space, and the flow of electrons through these is going to power these proton pumps to create that super strong proton gradient, right, in this outer outer mitochondrial space. And then you will have then a series of these, so that's step 4, the electron transport and proton pumping. And then step 5 then is these ATP synthases utilizing the flow of protons back downhill, right, from the outer mitochondrial space, flowing through them back down into the inner mitochondrial space. These are the turnstiles, and this is the crowd of people coming through. They are now making a ton of ATP in a mass production kind of way, right? So next lecture, we will look at how this electron transport pumping works and the step 5, the ATP synthesis. But for the rest of this lecture, let's focus on the citric acid cycle and understand it better, right? So remember, it begins with the oxidation of pyruvate to acetyl-CoA. So acetyl-CoA is the entry point for the citric acid cycle. Let's look at the structure of this here, because I think this is very enlightening, right? So here on the left is pyruvate, the end product of glycolysis. You might not have realized this, but when you look at pyruvate, right, so it has the 3 carbons that represent half of the carbons that came from the original glucose, right? The original glucose has 6 carbons. They were rearranged to be kind of symmetrical. Then they were broken in half into 2 3-carbon intermediates, like the glyceraldehyde 3-phosphate, that then each proceeded through glycolysis, right, and got worked on. So each became a molecule of pyruvate, right? So each has 3 carbons from the original glucose on it. But if you look at this, right, this carbon with the 2 oxygens attached, that's carbon dioxide. Right? The other remaining 2 carbons, hey, this is an acetyl group. Right? From those of you who remember your OChem, right, this would be an acetyl group. So there is an enzyme known as coenzyme A. This is the structure up here. Okay, so you can see it's actually a nucleotide, right? So you can see this familiar sort of appearance of a nucleotide, right, with this chain. attached to it. This is called coenzyme A. What coenzyme A does, right, is it comes along, there's an enzyme that will bind to coenzyme A as a substrate, and also to the pyruvate as a substrate, right? And it'll break the carbon dioxide off of the pyruvate, so the carbon dioxide is just released. That's a waste product of this. The remaining acetyl part, these two carbons, get added on to the coenzyme A, and so here it does appear. Right? This is the acetyl group. And now you have what we call, acetyl-CoA. Right? Now you understand where the name comes from. And so you have the two carbons with this CoA attached to it. Right? And this is what's going to feed into the citric acid cycle. Importantly, this is also an entry point into the citric acid cycle of other metabolites. So for example, proteins that are being broken down can be converted into acetyl-CoA. Fats like triglycerides can be broken down and converted into acetyl-CoA. We often think about fat and the metabolism of fat, of tapping into long-term stored energy, right? Because if you eat a lot, like I do, right? And you really don't need to have all of this glucose, all these sugars and things like that. Your body doesn't want to just get rid of it because evolutionarily, I don't know, when the next time I'm going to have a big meal is, right? At least that's how Mother Nature designed us to be prepared. We don't want to waste it. So if I really don't need all that extra sugar, it can be converted, first into glycogen. But if I'm not even going to use the glycogen, right, then it can be converted into triglycerides, save it for a rainy day. So those triglycerides then can be broken down, right, when I need extra energy. Right? When I say extra energy, I mean what? Extra ATPs. Right? So the breakdown of triglycerides can enter the aerobic respiration pathway over here. Right? But of course, it's much easier to just start with sugar. So that's why we often, lament the fact that it's hard to burn our fat, our extra fat, right? Because the problem is your body would always prefer, the easy way of doing it is just to take glucose, go through pyruvate, and feed it into the citric acid cycle. Right? It's less involved. Anyhow, so the acetyl-CoA feeds into the citric acid cycle. And you can see in this process of making acetyl-CoA, you not only lose a molecule of carbon dioxide, but you also fill one of these NAD electron carrying baskets. All right. So here is the citric acid cycle. All right. So I can hear this big thump as all of you are like falling out of your seats, right, in front of your computer. Don't worry. You don't have to memorize it in this form. All right. So why do I show it to you in this form? Because I think this cartoon does a really nice job through color coding things to help you understand, right, how the citric acid cycle actually works. Right? So up until now, you may have thought, oh yeah, the citric acid cycle is all about taking the pyruvate, you convert it into acetyl-CoA, and now you're going to really break it down to get all the carbon-carbon bonds apart, right? So you imagine that throughout the citric acid cycle, you see like, right, the half of the glucose molecules really being taken apart now, but you don't actually see that, right? And that can be kind of disappointing and very confusing, right? Look here. Here we have the pyruvate at the end of glycolysis being converted into acetyl-CoA, right? That's what we just studied. That's what we just looked at. And the acetyl-CoA is then fed into the cycle, right? It joins up with this intermediate molecule here, this oxaloacetate. The color coding, again, is nice because what they've done is in gray, they've highlighted carbon dioxide groups that are then going to be subject to being removed, right? And they've also highlighted in pink, right, the starting carbons that sort of help you keep track of, oh yeah, these two carbons came from the original glucose that went through glycolysis. Right? Remember, the original pyruvate is actually 3 carbons, right? But one of them was already the CO2 that gets lost in the conversion of pyruvate into acetyl-CoA. So you're left with these 2 pink carbons that then enter. You can see here it is. They are added on to the oxaloacetate to form citrate. And then it's converted through different enzymes. So this CAC1, 2, 3, et cetera, are the different citric acid cycle enzymes. Right? And so the citrate gets sort of reworked into isocitrate, and then you begin to break off these carbon dioxides. Right? So this group, this gray group here breaks off. Right? So that's what's represented here. And in that process, you free up some electrons. So an electron carrying basket gets filled up. So you end up with this ketoglutarate here. Right? So again, here's the original 2 carbons that came from glycolysis. And now you're left with 1 carbon dioxide on this big intermediate molecule. Right? This carbon dioxide gets broken off again, or not again, but again, another carbon dioxide gets broken off. So that's released here. And in the process, you fill another electron carrying basket. Right? And so on and so forth. But we can see from this point on, it looks like you're just kind of rearranging the bonds here. Right? And that is essentially what you're doing. This one step between the succinyl-CoA and succinate steps is the step where you actually can have this enzyme make 1 molecule of ATP. Right? But that's still, not the main purpose of the citric acid cycle, right? That's just a little side benefit from it. Look at what happens to the pink carbons, right? Suddenly between this step and this step, it's like they disappear, right? They treat it as though it disappears because now you're forming a double bond with this carbon to this one here. So now the behavior or this functional group that you've treated as a group that was added on in the first place, and that's why they kept it pink up until now, right? Now has really just become part of this fumarate structure, right? And notice what has happened. This now becomes a carbon dioxide that later on, as you convert this to oxaloacetate, becomes one of these gray carbon dioxides, right? And likewise, this group down here, right, becomes another gray carbon dioxide, right? And so that's why this thing keeps going around like this. So you might say, well, that's really disappointing, right? Because I don't see how the energy from the glucose is being tapped into, because the pink bonds just seem to magically disappear. Think about it this way. The carbons that are being contributed by the starting glucose molecule, right? These carbon-carbon bonds essentially get absorbed into the structure of these big intermediate molecules, but it's like they take their turn, because eventually after 1 turn of the citric acid cycle, right, these pink carbons have become the gray carbons, right, that are then lost as carbon dioxide, right? So you're continuously feeding carbon-carbon bonds into this, and you're continuously rearranging and breaking carbon-carbon bonds to get all these electrons out of it. Right. One way to think about it is this. Right. Carbons are floating in and out of the citric acid cycle, so you don't really appreciate this net change. Right? That's why it's hard to appreciate what the citric acid cycle is really doing. Think about it like soup cans at the supermarket. Right? Let's say you have the owner of a small little market. Every morning he restocks his shelf. Right? So he goes to the back room where he has his pallets of Campbell's soups, right? And then he takes them out and he starts loading them on top of the shelf to keep it nice and full. Think of these soup cans as the carbon-carbons, right? Or the acetyl-CoAs that are coming in. As this is happening throughout the day, you have customers coming along and randomly taking other cans, right? It's not necessarily the cans that he put on this morning, because he just did that this morning to keep the shelf stocked, right? But over time, you have customers taking cans out, and then he replenishes more. So if you came back a day or 2 days later, right, and looked at the shelf, you'd be like, oh, nothing's changed. It looks full, right? So I don't get it, right? I don't understand this whole citric acid cycle thing, because carbons are supposed to come in and go out, but nothing changes, right? It's because it's continuously this in and out. Think about it this way. If he ran out of inventory of soup cans, right, eventually he cannot restock. And eventually the shelf would have fewer and fewer cans, right? And eventually there'd be the last customer who comes and takes the last can, right? What happens then? Well, he's in trouble, right? So think about it this way. If you stop getting glucose into your cell, right, that gets broken down to pyruvate and then acetyl-CoA, right, you cannot continuously feed the citric acid cycle and eventually all the carbons, right? You don't replenish it, and the cell can no longer make ATPs through aerobic respiration, and the cell will die, right? So think about it that way. So this is the very last slide, right? This is the more simplified way that I want you to be able to remember the citric acid cycle. So instead of 8 separate discrete enzymatic steps, I think I have 6 different intermediates here. And the way that I've shown them is to remind you about how many carbons are part of these intermediate molecules. So in other words, oxaloacetate, right, has 4 carbons in it. it joins up with the acetyl-CoA that's coming in to the citric acid cycle. So 4 + 2, you end up with citric acid. Right? And then as you go around and start breaking the carbon-carbon bonds from 6 carbons to 5 carbons to get ketoglutarate, that's when you're freeing up these electrons that are going into these electron-carrying baskets. Right? You have that one step over here where you can make a molecule of ATP from it. Right? But that's not what the citric acid cycle is about. It's about making more of these filled electron-carrying baskets as you go around and eventually end up with oxaloacetate again, right? That joins up with the next acetyl-CoA. So each turn of the cycle generates 1 ATP directly, but importantly, 3 high-energy NADH baskets carrying electrons and 1 of these lower-energy FADH2 baskets carrying electrons. And we'll talk about what we mean by high energy versus low energy in our next lecture. Okay, so be familiar with this slide, but use the more complicated one with the little color coding to really kind of understand that soup can analogy of things going in and things going out. Okay, all right, let's go ahead and stop there.